Rational drug design is the process by which scientists shape a molecule from the ground up to make a potent, safe, and useful medical treatment. The projects to bring these materials to life are substantial undertakings. These massive endeavors combine and challenge the creativity and ingenuity of all the researchers across the project, from chemists to biologists to medical doctors and increasingly to computer scientists. In this two-part episode, we'll explore the mechanism and design of Gilead's drug Remdesivir as it pertains to the COVID-19. In this video, we'll talk about how Remdesivir actually inhibits the SARS-CoV-2 virus. In the next, we'll talk about how other factors led to its final design. Before we get to Remdesivir, though, let's talk about how we got here. The 2019 to 2020 outbreak of the SARS-CoV-2-based infection COVID-19 for coronavirus disease 2019 is not the first large-scale coronavirus-based disease in humans. It's not even the first type of coronavirus disease on which Remdesivir has been researched. However, as of the publication of this video, it's the most immediately pressing, so it's the one I'm going to focus on here. Coronaviruses belong to a larger class of viruses called positive-sense single-stranded RNA viruses. This larger class includes a number of other viruses, including West Nile virus and the rhinovirus that causes the common cold. It's also something of a mouthful, so let's break down what it means. Well, you've probably heard of DNA, deoxyribonucleic acid, and how it's the blueprints of a cell. You've also probably talked about the alphabet of DNA, ATCG, which stands for adenine, thymine, cytosine, and guanine. These letters, technically they're called nucleobases, are attached to a type of sugar called deoxyribose, which is then attached to a phosphate. The whole structure looks like this, though for the sake of simplicity, we often abstract it as a cartoon. For a given type of nucleic acid, like DNA or RNA, the variable part is the nucleobase and the rest is actually consistent. The deoxyribose and the phosphates bind alternatingly to make a long chain that makes up the backbone of the DNA, and the letters hang off the side like the rungs of a ladder. Two carbons on the sugar, carbons 5' and 3', are the sites of attachment for adjacent phosphates. DNA is usually double-stranded. If one side has an A, the other side will have a T, and if one side has a C, the other side will have a G. Note that this means that if you know what one strand of the DNA is, then you also know what the other strand must be. This idea is called complementarity, and the two matching strands are called complementary strands. This means that the two strands provide redundancy and not extra information. This double-strandedness forms the famous double helix structure that every movie ever gets wrong. It looks like this, not like this. I don't know what this is. Usually, only one side of the DNA in any given region actually gets read by the cell. Again, the other side technically has the same information content, but it's written as the exact opposite of what the cell wants to read. We call the nucleic acid strands that have readable information the sense strands, and the other strands the antisense strands. When your cell reads DNA to make a protein, it doesn't actually read the DNA directly. Instead, it makes a transcript of the DNA using a different kind of molecule, RNA, or ribonucleic acid. The differences between these two nucleic acids aren't huge. RNA has an oxygen in it that DNA doesn't have on carbon 2'. This lack of an oxygen makes DNA more stable than RNA, since certain enzymes and processes can use these oxygens as a handle to degrade nucleic acids. This is actually a good thing both ways. DNA is more permanent storage, and messenger RNA transcripts are meant to be more temporary. RNA also has a slightly different alphabet. Instead of thymine, we have uracil. It's also usually single-stranded instead of double-stranded, though there are many, many exceptions. After transcribing the information from the DNA, this messenger RNA eventually goes to organelles called ribosomes, where it gets translated into proteins. These proteins vary in function, from being structural units of the cell to being enzymes that catalyze reactions and everything in between. And because other biomolecules are made by proteins, regulating proteins lets cells regulate any number of biological processes. Okay, so let's take a look back at the SARS-CoV-2 coronavirus. Like I said, it's a positive-sense, single-stranded RNA virus. Let's take that apart. This virus uses RNA, not DNA, to store its genetic information. The RNA it has is single-stranded in the virus, not double-stranded, or I guess something even more esoteric than that. The RNA is also a coding strand, a sense strand, as opposed to an antisense strand that wouldn't meaningfully be translatable into proteins right away. The exterior of each coronavirus is made out of an oily layer similar to the membrane of a cell. This layer has a number of proteins embedded in it, including the S protein that gives the virus its distinctive shape. Inside the membrane is the viral RNA wound around protein-based spindles. When a coronavirus encounters a target cell, it binds and is brought in by hijacking the human ACE2 receptor, which usually helps regulate blood pressure. Once inside the cell, the RNA unwinds from the spindles and gets turned into proteins by the host's ribosomes. Some of these new proteins are structural proteins for a new virus, and some of them are functional proteins, called non-structural proteins because we're creative, that the virus needs to help replicate itself in the host cell. One of the non-structural proteins is an enzyme called RNA-dependent RNA polymerase, or RDRP. This protein is responsible for replicating the viral RNA to amplify the viral signal. This viral RNA then gets turned into other viral proteins by the host cell's ribosomes. After a while, some of these new proteins, some materials from the host cell itself, and a copy of the viral RNA come together to form a new virus, which can then go on to infect another cell. Given this mode of infection, there's multiple places we could imagine targeting a coronavirus. For instance, we could target its binding to the ACE2 receptor and prevent it from entering the host cell in the first place, maybe building up blood pressure medication research already targeting the receptor. We could also try to target the assembly of the final viral particle somehow, or even upregulate the patient's immune system to be able to kill infected cells. For Remdesivir, though, we're going to be targeting RDRP, and we're going to be doing it in a clever way. So this molecule is called ATP. You've probably heard about it in the context of being some sort of energy currency for cells. And it is, but it's also a building block for making new RNA. ATP stands for adenosine triphosphate. Adenosine, in turn, is what you get when you take adenine and you add ribose. RNA polymerases are enzymes that take molecules like ATP, GTP, UTP, or CTP and combine them to form chains, reading off another nucleic acid as a template as it does so. These new chains are new strands of RNA. Let's take a look at these precursors. Over on the right-hand side, we have the nitrogenous base, the letter of the RNA alphabet. This is the only part of these molecules that differs, so it's the part of the molecule that will be used to store information. Here we have a sugar, ribose, and here we have 3 phosphates, one after another. When the nucleotides are combined to form chains, 2 of the phosphates leave, and the remaining phosphate forms a bond to this oxygen here on the previous ribose of the growing chain. Keep that oxygen in mind, it'll be important later. This reaction is energetically favorable. This bond here is actually pretty weak, and we can make a much stronger bond when we substitute with a bond to this oxygen. Polymerase enzymes, like RDRP, are also good at making this attack by this oxygen particularly easy. Once a polymerase is done copying information, the new strand of RNA detaches and can go off to do whatever it is that it needs to do, such as give instructions to a ribosome to make a new protein. Normally, this is fine. Your cells do this all the time, and like we talked about, it's part of the process of how cells turn information in their DNA into proteins. However, if the information in that RNA came from a virus, not your cell, well, you probably don't want to make those proteins. They're probably not very good for you. Now, let's talk about coronavirus's RDRP, and specifically how we're going to target it. One thing we could do is give the RNA polymerase something that looks a lot like ATP or GTP or any of its other substrates, but which actually can't work to form a bond. For instance, if we take the ATP and replace this oxygen with carbon, we've destroyed the ability of these 2 phosphoruses to act as a leaving group, so no new bond is formed. And this strategy has been used in various labs to study processes involving ATP, including viral replication. We could also imagine getting rid of the oxygen here, so that if the nucleotide does get incorporated, then the next nucleotide can't be, and the message gets cut short. This is how the anti-herpes medication acyclovir works. Probably the next most relevant drug to remdesivir is sofosbuvir, which is also made and developed by Gilead and targets hepatitis C's RDRP. This molecule does have the oxygen it needs to form a bond to another nucleotide, but the oxygen is blocked off by a bulky methyl group, which is this carbon in the 3 hydrogens, and that tail jams up the RDRP machinery when it tries to add another pair. Remdesivir works in a similar way to sofosbuvir. This molecule is not actually remdesivir. It's the activated form of remdesivir, which the drug only actually becomes when it gets processed by a cell. We'll talk more about why that happens later. Let's compare this structure to ATP. Spot the differences? Well, there's a couple. First, you'll notice that we've changed up the nucleoside ring structure. Importantly, we've swapped out this nitrogen in ATP for a carbon in the remdesivir metabolite. Now, you might be thinking that we've changed how the cell will read this base pair, but we're not really. The cell actually reads this part over here, and we haven't changed that. That's important since otherwise RDRP might not mistake our molecule for ATP, which is what we want to have happen. What we're actually doing is making a more permanent carbon-carbon bond here in our drug, rather than the less permanent, more easily cleaved carbon-nitrogen bond in ATP. This less permanent bond lets ATP get recycled if it needs to be. And being recyclable is a good thing for ATP, but we don't want our drug to be recycled by the cell that would destroy the drug, so this substitution is a good idea. The actual active part of the molecule, where remdesivir is very, very sneaky, is actually right here. In the drug, we've added this extra carbon that's triple bonded to a nitrogen. This is called a nitrile, or a cyano group. You've probably heard of cyanide, and yeah, that's what you get if you have a cyano group on its own, and you might be tempted to think that there's some chemical similarity. But no, the actual mechanism of remdesivir is completely unrelated. So this molecule, the active remdesivir analyte, does look a lot like ATP, and RDRP does actually confuse it for ATP and can put it into a growing RNA chain if it's trying to reach for a letter A. It also actually can form a bond to the immediately next nucleotide just fine. What happens though is that after 3 more base pairs are added, the RDRP just stops making the chain. The current understanding is that the cyano group catches on the RDRP enzyme when the chain reaches this position. This jams up the RDRP machinery and stops the chain from being able to be replicated. There's another thing we need to think about with any drug, which is what the effect on a normal healthy cell might be. Remember from the beginning, you also need to make proteins, so you also need to make RNA. And the way you make RNA isn't completely different from the way coronavirus does. It's just a different specific tool that does more or less the same process. So there's a real concern that remdesivir might just get installed into one of your growing RNA chains and terminate one of those, which obviously would be bad. Luckily, your own cells don't seem to be bothered by remdesivir. The amount of compound required before the drug becomes toxic to one of your cells is extremely high, indicating that your own cellular machinery can deal with remdesivir just fine. And as it turns out, coronavirus RDRPs are particularly bad at telling the difference between the two compounds. These two facts combined indicate that remdesivir is likely to be a safe drug. Further research, of course, has shown this to be the case, and remdesivir is an FDA-approved drug for the treatment of COVID-19. Okay, so that's how remdesivir works. Well, wait, no, that's how this molecule works, but like we've talked about, that's not actually remdesivir. This is. Okay, so that just kind of raises a few questions. First, if this is the active form of the compound, and it is, why don't we just take that? Why does remdesivir have all this extra machinery on it? What is that doing? Second, how do we go from remdesivir to the active form? Well, in the next video, we'll talk about something called a prodrug strategy, one remdesivir follows. We'll also talk about how only considering the immediate target of a molecule doesn't actually necessarily result in a useful drug when you add the complexity of even just a cell, let alone an entire organism. For now though, thanks for watching and I hope to see you then.